So I'm Justin Schell. I'm with the libraries. I run DASH, Digital Arts, Sciences, and Humanities. It's an interdisciplinary project focused on emerging digital tools and methodologies for research, pedagogical, and artistic projects. And so we work with things like 3D scanning, 3D printing, data visualization, data sonification, text and data mining, things like that, and crowdsourcing, which is why you're all here today. And so I've been working with Lucia and with Zooniverse for the last 6-8 months now?
At least.
I don't know, something like that.
Maybe a year.
And so we wanted to put together an event within CFANS and CBS and start encouraging more projects in these areas as well. So I'm the sort of unofficial MC of this. And so just to give you a sort of roadmap of what we're doing today, we're going to have 3 presentations. First by Lucy Fortson from Physics and the Citizen Science Alliance, which is the parent organization of Zooniverse. We'll have a presentation by George Weiblen of the Bell Museum in Plant Biology and Keith Barker from, Evolution, Ecology, and Behavior. I was trying to get the EEB, the right ones in the right order, and the Bell Museum, about a project that is currently under development with Zooniverse, with plant specimens in the Bell Museum collection. And then Garrett Beier, a graduate student from plant pathology, will talk about a project that is just getting under development with Dutch elm disease and tree cuts. So after that, we're going to have about a 45-minute to an hour brainstorming session where we can explore your project ideas and how some of these tools through Zooniverse and different platforms that Zooniverse offers can be used for your own projects. And I'm really excited to hear what you have and what sort of bubbles up after these conversations. So I want to say a quick thank you to Plant Pathology and to Jim Berdeen and Shauna for helping organize this and getting everything together. Thank you to the libraries for also helping this out. And again, thanks to you for showing up today on this sort of cold, not so April day.
It's not cold.
It's not cold. It's, I could see my breath out there. That's a bad sign. So without further ado, I'll hand it over to Lucy Fortson. Thanks.
All right, thanks very much, Justin, and I'm very happy to be here, and thanks, Jim, as well. This is great. I am coming over to this side of the campus more and more and more, which is a really good thing because it means I'm really beginning to integrate into areas where the Zooniverse is trying to grow as well. So I am going to talk. This is the outline that basically Justin gave as well. But so I'm going to give the broad overview of the Zooniverse, how it got started, what it is, the various different types of projects that we do, the types of tasks that you might imagine the public doing for your project to help get the wheels going in your head about imagining what they might be able to do with your a project that you might be interested in putting up with the Zooniverse. Give you a little bit about the architecture of the Zooniverse and some changes that we're implementing right now, and then just wrap it up with some thoughts of how we can move forward together. Okay, so hopefully everybody knows what these are. Yay, okay. This is the participation part starts early, okay. We all know what these are, right? Okay. I don't even, we're on this side of the campus, so I don't even need to, okay. What about these? History, papyri, okay. Cyclones, or whatever, depends upon which side of the earth you're on, but yeah. Okay, but really, these are all images from projects that rely on the analysis of complex images and pattern recognition capabilities. And they all share in common the fact that computer algorithms currently are not able to do a full analysis of those images and give the researchers the information they need about the data. about those images. Okay, so we all see there's a tiger in the brushes there, but which is better at spotting the tiger? A computer? Okay, well, the answer is there. Of course, the 5-year-old child, hopefully even a 2-year-old child, is better at spotting the tiger than the computer, right? So computers are really, really good at numbers. They just outclass us, you know, don't even try, okay? So 1 billion operations per second. You could just, you know, click away on those numbers, you know, till the end of days. However, humans have developed by necessity, by evolution, the need to distinguish the tiger from the bushes. Those who did not are no longer with us genetically. So another thing that humans do is we like to classify, right? So we discriminate. Again, an evolutionary tick. Right? If we don't make the right decisions based on the information we have, we're also out of the gene pool. Okay, so we like to categorize things. Now we have the luxury, because we have a fairly advanced civilization, to be able to categorize galaxies. So this is what astronomers have been doing since Edwin Hubble, 100 some odd years ago, when he first looked at the first 100 or so galaxies from the Mount Wilson Observatory in California. And he found, and those following him found, that galaxies typically come in 2 basic types: spirals and ellipticals. Spirals tend to be blue because it turns out that there's a lot of star formation going on in them. Ellipticals tend to be red because, as it turns out, the star formation has ceased in most ellipticals. This will become important in a few slides. So just remember, blue for spirals, red for ellipticals. Historically, Edwin Hubble, he himself would go in and classify and categorize all the galaxies that he had observed into these different categories. In the 1950s and 1980s, when we were at the point of obtaining tens of thousands of galaxies on photographic plates, maybe the faculty wouldn't do it, but they would assign the graduate students to do the morphological classification. Right? But it's still possible for one so-called expert to be able to go through all of the tens of thousands of images of galaxies and do a morphological classification. The problem comes in when we get to the 2000s and the Sloan Digital Sky Survey, the first full sky survey, and they are returning millions of galaxies. Right? And the problem gets even worse when we talk about 2020, when the Large Synoptic Survey Telescope is going to be online, and we are going to be recording several billion galaxies. Okay, so at that point, you're not going to have any graduate student touching it with a 10-foot pole. Okay, so we have this issue. We have all of this data pouring in on us from all sorts of sensors around the world. Doesn't matter what discipline you're in. Okay, so you go back to those first sets of images. So much data is coming in from historical records, from ocean sensors, from satellites, from telescopes, from our smartphones. Okay, so it's just, we're wading and drowning in what we call the data deluge. And the issue then becomes, it's not just a question of storing all of that data, it's how can we keep up with it in terms of analysis. So there's a gap between our ability, okay, so we're detecting all this data, it's coming in, we're storing it, but we don't have the tools yet built to analyze data coming in at the volume, variety, and velocity, the terms that people like to use when they're describing big data. So we need to improve our computer algorithms to process that much data, of course. So then the question becomes, what is the best algorithm to process complex images? Okay? So remember, computers are really good at numbers. Right? So you can imagine that a lot of that data can be processed with computers if we're just talking about numbers. But what about telling the difference between the spiral arms and the elliptical bulge? Right? If every single galaxy itself was the same spiral pattern, perhaps you could write a computer algorithm to do that. But every single galaxy is like a snowflake. It's, its own character, its own fingerprints. Okay, so they're unique, and therefore it is nearly impossible to write a computer algorithm to determine whether a galaxy is a spiral or an elliptical. So the best algorithm that can process complex data is that. Okay, it is the human brain. Right? So we know that because we had graduate students and even faculty. Their brains may not be as good as the graduate students, but we had the human brain in the beginning doing the morphological classifications. So we know that the brain is the best thing to use at this point. The question is, how can we parallelize this, right? So how can we computerize, if you will, that best algorithm? And we do this through crowdsourcing, right? So you just hook up what I call human processing units, right? As opposed to the central processing unit of your computer, we hook up the HPUs, get them working in parallel on the various images, and at that point we are a crowdsourcing project. So the story of galaxy evolution and classifying galaxies by their morphology is the beginning of the story of Zooniverse. And we start with a project called Galaxy Zoo, which launched in 2007. And it starts because a graduate student by the name of Kevin Shawinski at the University of Oxford had been asked to, in fact, classify a large fraction of the 1 million galaxies from the Sloan Digital Sky Survey. He got through about 50,000 of them in 1 week, which is a lot. At the end of that week, he took his postdoctoral advisor, Chris Lintott, who now happens to be the head of the Zooniverse, the PI of the Zooniverse. They went out to a pub, because I guess that's where all British people solve their problems. And they talked about what they could do. At that time, in 2007, NASA had launched a project called Stardust at Home, which was getting the public to classify some images that NASA had returned from space. And so they knew about this project. They knew that 20,000 people around the world had been helping NASA with their project. So they figured, 20,000 people, yeah, we could probably get 20,000 people interested in classifying galaxies. And if we want at least 3 classifications per galaxy, right, that's going to be about 3 million classifications we're going to need. And they worked out some numbers, slower than a computer, of course. And they figured out it'll take about 3 years to get through the 1 million galaxies. But in the meantime, Kevin could be doing other things, like preparing papers and doing other analyses that would be of interest to him graduating. So in the end, Galaxy Zoo was wildly successful. And the project actually finished in about 6 months. But it was left running for about 1.5 years. And after 1.5 years, 35 million classifications had been done by 150,000 users. Okay? That's roughly 3 1/3 continuous, 24/7, 365 days a year person years. Okay? Can't get a graduate student to do that. So not only did this allow Kevin to go off and do other things while he thought it would allow him to go off and do other things. At the end of the day, he actually had to pay attention to a lot of the public that were on the site asking questions and whatnot. But that was a nice price to pay for the public actually doing a lot of the work for him. The other thing it did was, for the very first time, instead of relying on a few experts, it gave if you will, a probabilistic way to measure morphology for galaxies. So we now have, on the right-hand side here, this is the probability of a galaxy being a spiral. Right? So all of the votes that came in, 35 on average per galaxy, right? If 35 out of those 35 said it was a spiral, it's going to land in this bin here. So 100% probability that it's a spiral. If 0 out of the 35 say it's a spiral, then it's 0% chance that it's a spiral, but because we're in a binary system, it's 100% chance it's an elliptical. All right, so the point here is, depending on the research you're interested in, you can pick what cut you want to make on your data sample at this point. So you can say, I have a research problem which requires a very pure sample of spiral galaxies. So I'm only going to take this bin right here. But if if I can have a little bit of noise, if I can have a few ellipticals in my sample, I'm going to, maybe cut at 80% probability, just to increase the sample size a little bit. Okay. For the first time, we've been able to do what I would call even fairly simple data cuts on morphological classifications of galaxies, which is incredibly useful for studying galaxy evolution. Okay, so Galaxy Zoo was incredibly successful. At the time the Galaxy Zoo was running, we had many people writing to us asking, hey, can we do the same thing with our project? I mean, we had people from papyrology to crows, looking at crow behavior. I guess they're New Caledonian crows or something like that. They use tools in a specific way, and there's a lot of video archives of them. That project, for whatever reason, never got off the ground after 8 years. So if you have a crow project, we'd love to. It was one of the top ones on our list to do. We haven't done it yet. So, so we knew that there was an appetite from the researchers out there. What we didn't exactly know was what the appetite was for the crowd. Was it that the crowd really liked pictures of galaxies? So we started preparing ourselves for, what do we need to do to build an infrastructure like the Zooniverse? So we took a look at the variety of types of citizen science that were out there. And we categorized them. Again, we want to categorize things. So there was this sort of passive type of citizen science that some of you may know of. These sort of, when you download a screensaver like SETI at home, you just let it run and fill the CPUs with, when you're not working on something. But this does not engage the human brain as much, except for just that point when you're actually doing the download. The data collection piece of it is sort of the classic version of citizen science. And this is, going all the way back to so-called, gentlemen scientists who are wandering around on their estates and finding fossils or, collecting, samples of trees or what have you, or amateur astronomers, or, with the Christmas bird count from the Audubon Society and this sort of thing. So this is sort of the classic type of citizen science that we might have thought about before the internet. The internet really enabled it's interesting, I jumped from 2 to 5. Don't know what's missing in between. Maybe we'll figure that out eventually. But so we have this sort of data analysis part of citizen science that has been enabled by the internet. Okay, so as I'm saying, this sort of crowdsourcing aspect where you can chunk out the data analysis process and send it out to a bunch of HPUs out there in the world. Okay, and there are many examples of this kind. And so Zooniverse was clear, was going to exploit this particular niche. Right? So one thing we do not do, just to be very clear, Zooniverse does not do any work with data collection citizen science. That is something I would personally like to change, and I would like the University of Minnesota to be part of that change. So if there are data collection projects that you work with, I would like to talk with you about how we can link the data collection side to the data analytics side through a combination of whatever it is that you're doing with the Zooniverse. But before we actually built the infrastructure, we needed to answer this question, would the crowd be interested? And we also needed to answer the question of, all right, they might be interested, but how much time do they actually have to give over to crowdsourcing? So we did a motivation survey, and we found amazingly that 40% of the people said they wanted to contribute to research. It wasn't the fact that their teachers made them do it. It wasn't the fact that they thought galaxies were beautiful or they had a prior interest in astronomy. Those were all contributing factors. But the largest factor by far was they wanted to contribute to real research. That floored us. Okay, these are just members of the public participating on Galaxy Zoo. Okay, so check. That's good. We've got lots of research for people to participate in. So then the next question is, by the way, that was 20,000 people that we asked that question of. The next question is, what is the cognitive surplus? All right, so this is Clay Shirky has this book called Cognitive Surplus, and then he has this video that goes along with it. And so these are Google boxes. This is 5 years old, as it turns out. I'm sure it hasn't decreased. But as of 5 years ago, US adults were spending 200 billion hours a year watching TV, as opposed to this little dot over here, which is 100 million hours it took to create Wikipedia. Those are both completely outclassed by Angry Birds, okay? So my son says this slide is pretty old. It's now, whatever, Clash of Clans or something, I don't know, whatever, some other game. Okay, so we've made several points here. We know that we want to tap into that cognitive surplus, and we know that the people out there are very interested in contributing to research. So we've shown that the scale of the problem, sorry, this pointer is dying, but the scale of the problem we've discussed. You know, certainly in astronomy, we have this data deluge coming in, and I know it's true across all disciplines. We now also have the ability to provide measurable accuracy on problems like classification. Okay, so we don't have to rely on experts and potentially their blind spots. Okay? Another thing that I won't go into, I'll touch on a little bit later in the talk, is education. So you put this stuff out to the public, education comes along for free, effectively, right? So, you know, obviously that's not a completely true statement. You have to work to, if you really want to get projects into a classroom, yes, you have to do a lot more work. But in terms of sort of outreach and educating the crowd about the process of research, we actually have evidence that the Zooniverse projects are doing that. So there's another key piece, which is machine learning. So I mentioned before that computers are not very good at these discerning these complex patterns and images, but they're not far behind, okay? So there's a lot of computer scientists who are working very hard to develop algorithms, for example, for facial pattern recognition, for face recognition. And so one of the things that we actually say is a sign of a successful Zooniverse project is if they close themselves down, because they've developed an algorithm based on what the HPUs have provided as training data to improve the computer algorithms. Don't worry, the citizen scientists aren't going to be put out of a job. There's always more stuff because remember, we have that data deluge, okay? So this is this human-machine partnership we like to talk about. We have the data deluge coming in. The ever-improving machine classification routines are going to get through 80% or so of that data. All the stuff that the machine algorithms can't do, they're going to pass off to the citizen scientists. The citizen scientists are going to do the classifications. Those classifications form the training set for the next round of improvements on the machine algorithms. And at the end of it all, in combination with humans and machines doing what each is best at, you get the best set of information and knowledge and understanding out of it. So we have implemented this cycle in a couple of projects now, most notably Supernova Zoo. And it also enables us to study how the citizens, to do some sort of sociological studies on the accuracy of citizen scientists, but that's a whole other story as well. So finally, I wanted to touch on serendipity, the idea that we can find the unknown unknowns. So there's several classes of knowledge. The known knowns, okay, we're going back to Donald Rumsfeld here. These are the primary tasks. Okay, this is the data reduction done by the science team. Known unknowns are theoretical possibilities, but where they actually haven't been discovered. The objects actually haven't been discovered. And they can be discovered in the data set by the public. Then we have the sort of completely unknown unknowns where the human brain has the capacity to be presented with an image that doesn't look like anything they've seen before and ask that question, wait, what's this? And then, proceed from there. So several discoveries have been made with this, several unknown unknowns. In fact, an entire class of galaxies called green peas, because they were discovered by the public. They look like little green round peas. So the public called them green peas. So officially in astronomy, they are now called green pea galaxies. So, so the Zooniverse was launched in early 2010. Over 1 million volunteers, we're about 1.3 million volunteers worldwide participating in real research. It's a part of the solution to the big data problem. We've worked on various types of data products, images, simulations, text, videos, sound clips, you name it. We've probably done it, though don't push me on that, or actually do push me on that. If you have a completely different type of data format, I'm interested. We've got about 35 projects and always growing. We're going to launch very shortly something called Chimp and C. Okay, behavioral studies of chimpanzees. That's coming soon. Watch for it. We have in the disciplines over 60 peer-reviewed publications. We have citations that are in the several hundreds now. We are hosted on Amazon Web Services. The idea is that we occasionally have events such as associations with Stargazing Live, which is a big British TV program, and we can scale up to 100,000 events very quickly because we're hosted on, did I say events? 100,000 people quickly because we're hosted on Amazon Web Services. And we have funding from the National Science Foundation, Sloan Foundation, Google, the Levery Home Trust, NASA, Microsoft, a couple of others. And as Justin mentioned, we're operated by the Citizen Science Alliance, the member institutions listed there. So when I arrived here at the University of Minnesota in 2010, from the Adler Planetarium, where I was one of the co-founders of the Zooniverse, one of the very first things I did was, hey, you got to get the Zooniverse here, embedded in the University of Minnesota. It's a fantastic tool for researchers to be able to use. And since 2010, we've had 4 projects that represent the University of Minnesota across the various colleges. And so since 2010, I've been working with various deans of various colleges and other units, the OVPR, to try to pull together some funding to really enable multiple research projects to come through the University of Minnesota to Zooniverse. That was ended up being successful last November, and we have since hired a developer. We have a couple of projects, which we'll hear about a little bit later, that were part of the first round of a solicitation for proposals to come in and will be developed by the developer hired by the funds. So the point here that I'm trying to make is this is your money, okay? Make use of it. I'm, the keeper of it, but please, you should feel that the Zooniverse is a tool that is now here at the University of Minnesota that you should make use of if you can. And we would like to help you do that. So I'm going to speed up a little bit here. So the Zooniverse has grown a lot. You can see that each time we launch a project, there's a spike in the number of registrants, and then the cumulative registrations, of course, continue to accumulate. This tells you the value of even a small number of clicks. So this is a visual way of representing that each box is one volunteer, and the size of the box is the number of clicks, the number of classifications they have done. In this case, it's our old weather project, which is the number of transcriptions that has been done. So then by definition, the total area of the page then is the total number of clicks. Right? So if you just split it in half, you can see that roughly half of the clicks are coming from power users, and the other half are coming from people who log into the site and do maybe 3 or 4 transcriptions and disappear. Right? Turns out these are really valuable. Not just because they make up effectively half of the total transcriptions, but because in this process, we want multiple independent classifications, right? Not to say that these guys aren't useful, but we can use them in different ways. We know we can measure the accuracy of these guys at the end and use them as sort of baseline. Okay? So these become sort of our experts. These become our independent multiple classifiers. Okay, so the various types of projects. I mentioned a couple. We have a variety of tasks, a variety of output data types. One of our jobs when we set up a project is to elicit from the research team what kind of task, what is it that you exactly want the public to do? Okay, you have to narrow it down to a very, I'm not going to say easy, but accessible task or workflow, right? A set of tasks. And that then depends a lot on the kind of output data you're looking for. Once you get the output data, you have to apply a consensus algorithm. Say you get 35 votes on each galaxy. Well, you need to figure out how to combine those votes, okay, to give you a meaningful interpretation of the data. And then I mentioned these before, so I won't spend too much time on that. So this is just a very simple cartoon of how the infrastructure on Zooniverse works. We have a classification interface, which is what the visitor sees. They go in and they interact with this classification interface. They do the primary task. The clicks from that primary task go to a classification processor. That classification processor can then count if they have reached, it passes it to a retirement system. That retirement system, in the simplest cases, is hard-coded to say something like, we believe, the science team believes, that if we get 10 classifications on this particular image, we can say that image is finished because we have, for whatever reason, we believe that to be true because we've done some research ahead of time. You can make a much more sophisticated loop here, and I'll show you an example of that, where you can have a dynamic retirement system whereby you can actually measure whether or not you have what those probabilities of completion are. So for example, if you had 90% agreement on something, and if your research says 90% is awesome, then you can just retire that subject then and there. So you can do much more sophisticated algorithms here, but you can just go with the simple hard-coded, 30 or 80 or what have you. So once, if the subject then passes through the classification processor and says, nope, you're not ready to be retired yet, gets dumped into the pool, and then randomly selected again to be presented to the next user in the classification interface. If it is retired, then it goes into, potentially in the more sophisticated loops, you can actually make the consensus engine part of this. But once it's retired, it gets dumped into the consensus database, and a consensus algorithm is run on it to give to the scientists the output data. Okay, so let's go through several task types, and I'll try to pick this up. So the simplest, of course, is a decision tree. Galaxy Zoo was effectively a binary tree. There are a couple of other little additional questions, but it was effectively a binary tree. This is probably the most complex you would ever want a decision tree to get to. There are 37 different endpoints on this decision tree. Once you get to that level, then you probably want to think about moving to something like snapshot Serendipity, which will come up in a second, in which we put in a filter, actually put in a combination of decision tree and filter system. The weighting that you can do, again, either you can just take the straight average of the votes and go with that, or you can do much more complicated consensus algorithms where you go through several iterations, you find the users that are most accurate, and you use them as a weighting, and you compare everybody else to them. You cut out the bottom 20%, say, and you continue sort of iteratively until you get to a level of purity that you're interested in for your data. So there's, again, the consensus algorithm is what the science team wants, effectively. It's their job at some level to develop the consensus algorithm based on their research criteria. So another task is marking. Right, so this is space warps. It's searching for gravitational lens. So the idea here is you literally set a mark on the screen. And then you can do several things there. You can ask questions based on that mark that the person can click yes or no. So that would then be a combination of a decision tree and mark. Right, so if you're combining workflows here, right, you're saying I first need a mark on a page, which then in my consensus work, I can say, in an XY grid that I can then overlay on my image. Okay, there's a gravitational lens here. And I can relate that back to the actual object that is sitting in the image at that particular location. So marking locations on a page, and then potentially doing questions based on that marking. It turns out Space Warps uses a very sophisticated consensus algorithm, so it's probably our most sophisticated one yet. And it's a dynamic process in which it goes in with an a priori probability of 10 to the minus 4 of a classification actually being a lens. And then you kind of follow a trajectory here, and typically as any individual does a classification, whether it's a lens or not, bumps it one way or the other in terms of the probability, And most, of course, because they're not going to be lenses, are going to pretty quickly find themselves over here with past this rejection threshold of 10 to the minus 7 probability. Right? A few of them are actually going to end up getting bumped, kind of like a pinball. Not quite. It's more deterministic than that. But you end up over here where they've passed the detection threshold of 95%. Right? This is continuously updated. Each individual classifier on the system moves that subject one way or the other. And then once one of the thresholds, rejection or acceptance threshold, is passed, it's then retired from the system. And that is updated every 24 hours. We're actually moving to update it as we fly. So I also mentioned we can gain information about the accuracy of each individual classifier. So, I'm not going to go into the details of how we do that, but, Amazon does this, right? So, it's basically using some of the same algorithms. And yes, we do let people know that their information is being, retained. Okay, so we can do marking and measuring. So this is Plankton Portal. So we can, this is probably of great interest to this crowd. The idea that you can mark a location and or mark an object on a page and measure. There's several potential measuring tools that we can use, but the most common that we use is sort of semi-major, semi-minor axis measurements. You can, again, combine that with a decision tree if you want to ask questions about the measurements, or potentially annotation as well. All right, so then we have transcriptions, which are structured. So this is the Operation War Diary. These are officers' logs from World War I on the Western Front. And here we ask the public to fill in structured data from the logs. So it's not a full text transcription. They're reading the text and finding the information on the page that will then make it searchable and indexable later. So another prime example for this audience is the Notes from Nature project, which is another structured transcription project. And it's very similar to the project from the Bell Museum. So I'm actually going to not take too much time going through this. But suffice it to say, we have several projects with multiple collections under one framework in Notes from Nature. The idea, again, is to collect the metadata from the identification cards that are associated with each member of a collection. Those are then preserved as records that are associated with those images. The consensus algorithm for transcriptions is somewhat different than for image data. We have issues of completeness for transcription records. Here you have three potential outcomes. You can have completely complete, you can have partially complete, or you can have fully incomplete. Then layering on top of that, you also have accuracy. Okay? So depending upon the research you're trying to do, you either care more whether something is complete, because then an editor can go in and make it more accurate, or you actually care more about the accuracy because you have billions of things and you really just want accurate stuff, as much accurate stuff as you can get. But you can, toss the other stuff out, right? So you have to ask yourself the question, do you want completeness or do you want accuracy? Obviously, ideally, you would like both, but that's not always possible. The other mode for transcription is full text. This is a distinct example of full text transcription where we actually have people go in and fragments of papyri and classify morphologically each Greek letter in the fragment using the keyboard here. So you don't have to know Greek. You just match the pattern of the letter in on the fragment to the letter that best fits it on the keyboard. And in this way, we've transcribed, we've had millions of transcriptions now of fragments from ancient Egypt, as it turns out. Here, again, the consensus is a bit difficult. We first have to cluster all the little points. So this is a marking project, a marking and annotation project. Okay, so each mark is where an individual decided a letter's center was. So then we have to apply a kernel density estimator to estimate the centroid of the clicks. And then we have to ascribe a consensus letter to that particular location, right? We also need to provide the minority reports, because while you can provide the consensus letter so that you can produce the curated text here, you need, the raw consensus text, you want to be able to have as a final output a curated text so that the editor would go in and say, you know what, I like that minority report better. It actually makes more sense, okay? So this is a editorial tool that we've created on top of the consensus algorithms here with the MSI, Minnesota Supercomputing Institute. Okay. And that was funded by Minnesota Futures Award. Okay, so then we have, when you have this ginormous decision tree, actually sometimes it's better to go with filters. The reason is that if you force somebody to go through an entire an entire decision tree process, but they actually know that this is, let's see, what is this? An impala or a Grant's gazelle? Thompson's gazelle? Thompson's, okay. It's been a long time since I've done it, okay. Right, if you know that, and you were forced to go through an entire decision tree to get to that point, you'd be out of there. Okay? So you need to be able to give people the opportunity to click exactly what they know when they know it. At the same time, if you don't know it, you need the decision tree, right? So you can then click on different patterns, color, horns, tail, bill, whatever, to help you filter down to where you then get a few choices in a decision tree. So that's what I mean by filtering combined with a decision tree. So that output then is, of course, used to in ecological studies of the Serengeti and mapping. There's all sorts of research that's being done with that. And this is a great Minnesota project because the two graduate students working with Craig Packer on this, who devised the project and carried it out, Ali Swanson and Margaret Kosmala. Margaret is now at Harvard doing a citizen science project. And Ali Swanson is now at Oxford working with the Zooniverse. So, very successful project. We can also do comparisons, in this case, comparison of sonograms. Right, we have another sonogram project with bats. And here, the object is to try to identify dialects in the whale song. So you just have to match both orally and visually the sonograms. Okay, I'm nearing my time, so I think one of the key messages to get across now is that up to now, we have had to build every single one of those projects custom built. We have had to reject many, many excellent project proposals because we don't have the developer capacity to develop as many projects as we get requests for. So, and this includes projects that are potentially containing sensitive information, so it might need to be behind a firewall for privacy issues, so health, medical type projects. These also are projects that may have small numbers of images. That wasn't, for a cost-benefit analysis on our part, it wasn't worth the development effort if it was going to be finished so quickly. But they're still beautiful projects and completely legitimate projects to be using this type of tool. So we were motivated and thankfully received about $2 million from Google to develop a whole new technology stack that we're calling Panoptes, which ideally, when it's all said and done, will be as simple as WordPress if you want to actually launch your own project. Right? So that means there will be a set of templates. You'll pick the workflow. You'll construct the workflow based on the various things we just discussed. You'll figure out, it'll help you through figuring out how to ingest the data, and there will be a simple consensus algorithm that can be applied to it. You can launch it, you can put it behind a firewall, it can be on your own server. It can even potentially end up on the Zooniverse public site, depending on how things work out. So one of the things that I would like to do with the University of Minnesota, and in particular you guys, is find a set of beta testers for this new technology stack. And Garrett is hopefully going to be one of those beta testers, and he'll talk about his project in a few minutes. But we're basically mostly finished. We're debugging the technology stack, and now we're in the process of finding guinea pigs who want to exercise the process. So, we can talk more about that in a bit. So also in the infrastructure, we have basically mission control. We keep track of how the performance of the projects. One thing we found is that if science teams or research teams are not involved, that causes them, this is the average time on site and the average, and the total number of visitors. So we have projects that just aren't healthy for a variety of reasons. So we work to try to get the research team more involved, or if it's a horrible bug, obviously we're going to try to fix that. But we find that there's sort of a cluster here of fairly healthy projects that just kind of bump along getting their stuff done. And then you've got the projects that are up here. Like the Andromeda project was another University of Minnesota project. 1 million images from the Andromeda galaxy from the Hubble. We thought it was going to take a while. It took 10 days to finish, okay? Go figure, okay? So we're constantly monitoring the health of the projects and the loads on the systems. And it looks like a NASA control room, when you go to Zooniverse headquarters. I'm going to skip these 2 slides. This is basically the cartoon of what we're going for with Panoptes. Just finish with the idea that we actually do have an ethics of citizen science. We never want to launch a project that is not a research project, because that is, Zooniverse is a research tool. So we don't accept education-only projects. Panoptes is a perfect opportunity for somebody if they want to build an education-only project. So if they want to develop a lab or something like that using information that they've got and use this tool, perfect. And it also has to be a research project which can prove that machine algorithms don't yet work on that particular research problem. We must keep the volunteers informed of the progress and the process, and we must acknowledge the volunteers in the publication. And we typically do that by putting a link in the publication to a website that lists every single person that has contributed to the work. So along with the primary task, we have something called the talk tool, which is a way for the, they call it themselves zooites, the volunteers, to talk amongst themselves. This is where a lot of the discoveries are made. Right? So because the science teams can't pay attention to everything that is being said, we have found that the zooites actually work together to determine whether they think they've made a discovery or not. There's also blogs that we expect the research teams to sort of continuously update. It's another way of engaging the public and the volunteers. And as I said, we really noticed that if you don't have an engaged science team, the project will likely fail. There are several data analysis tools that have been developed for several of the projects, which then allows the more advanced users to help them make discoveries. Okay, so basic plotting and histogramming tools and mapping tools and things like that. Those tools have then been used in a particular project called Galaxy Zoo Quench, where the public has been involved from defining the research problem, classifications, the data analysis and discussion with the tools, and then actually writing the article for publication. So this was really, really ambitious. By the end, we had several thousand starting. By the end, there were 3 people that were still in there, but they were, they're, like a secretary and a lawyer and these are people whose interest kept going. Of course, we're using these tools and embedding them in particular in the University of Minnesota into introductory classes. For example, we're using Snapshot Serendipity in some of the biology classes, and we're using Galaxy Zoo in some of the introductory astronomy classes. And so I'm just going to leave that summary up here. The bottom line is we're moving into an era of do-it-yourself zoos with the launch of the Panoptes API. This is, I've said most of this, but this is the key piece here. Our first focus was on transcription projects, aka humanities, although it turns out that we selected a not exactly humanities project. That's fine. It's a transcription project. Our next focus is going to be on biology projects writ large. Okay, so we're going to be releasing requests for proposals soonish. I don't know exactly when, a couple of months maybe. But in the meantime, if you've got a project idea, talk to me, Justin, or Amy, or anybody that you think can get back to me, and we can talk. So I'll leave it there. Thanks.
All right, thanks, Justin and Lucy. I think we're just so fortunate to have Lucy and Zooniverse here at the University of Minnesota. So I'm really grateful for the opportunity to use this really exciting approach to doing science. And just want to briefly describe the project that Keith Barker and I are leading. Keith is here as well. I'll just be giving a very brief overview of our Zooniverse project, which, as Lucy said, came about through the call for proposals that was issued last year. Our focus is on the biodiversity edge, charting change where biomes meet. We're collaborating with the Zooniverse team, with the Minnesota Supercomputing Institute, University Libraries, Bell Museum. And our focus is on our region, and the idea that given our relatively highly seasonal climate and our unique history in terms of where we're positioned in the middle of the continent and what's happened to our biota here over Earth history, that this is a special place where very different kinds of ecosystems come together, the 3 major biomes of Minnesota. And these biomes being products of the history and the climate, we're poised here to experience environmental change and biotic responses to, say, climate change more rapidly and more dramatically than in many parts of the country. And we have the opportunity to document this change, try to understand what's happening, how fast is it happening, and perhaps predict where things are going. So shown here is a, let's see, this is the, shown here we've got a current distribution of a coniferous forest tree species. I think this is white pine, and here is a model prediction showing that that species is predicted to migrate north over time. As a natural history museum, the State Natural History Museum for Minnesota, designated by legislative mandate in 1872 to record and preserve the biodiversity of our state, we have records going back to the time of settlement on when and where organisms were observed. It's our mission as at the Bell Museum to discover, document, understand nature, promote informed stewardship of the natural world. It's a natural partnership for us to get involved with Zooniverse, given that the museum is very active in public engagement already. It's already doing the science of biodiversity discovery. We want to merge these efforts in a citizen science project that draws upon our rich collection of natural history objects. We have about 4 million objects in the Bell Museum collections, comprising many tens of thousands of species from Minnesota, our region, and the world. These specimens are they capture the history of biodiversity in our state. Presently, these specimens and their associated data are not readily accessible to the public. And so through the state legislature, through the program, which I'm sure many of you are aware of, the Legislative Citizen Commission on Minnesota Natural Resources, there are calls for proposals and awards are made through the Environment and Natural Resources Trust Fund. This is the lottery money. And natural history or natural resource data and information is one of the focal areas of these awards. We are in the process of receiving an award at the Bell Museum to integrate the Minnesota Biodiversity Record based on our specimens into a comprehensive and dynamic atlas. And we'll see in a moment how our partnership with Zooniverse will enable us to both engage the public and move this project along much more efficiently than we could if we just did it as curators in the museum with our graduate students. So the museum houses collections. It has databases that contain records of those specimens. We receive data and collections from various state agencies like the Pollution Control Agency, the Minnesota Biological Survey. Presently, only we're receiving that information, but we have not been serving it up to feed back to those agencies or to feed out to the public. And so the dashed lines here are what we're working on. This begins with generating digital images of the specimens and the associated label data. So this is an example from our plant collection, the herbarium. We have a press-preserved specimen here that has been digitally photographed in this contraption here, bar-coded, and there is a lot of label information associated with the specimen that we would like to transcribe into a database, and that's where Zooniverse comes in. The same is true for animal collections. where we have associated with the gopher. What is this? The 13-line ground squirrel. And here is collections information: the scientific name, the sex, the geographic location and the date on which it was collected, some habitat information, the name of the collector, the weight, and so forth. This is the kind of data that we have. It's rich data on the history of plant and animal distributions in the state that is presently incarcerated in the museum, and it's impossible to do any analysis on this. But the public and the state agencies are really keen on gaining access to this information to understand change. So this is the kind of handwritten label information that an optical character recognition software struggles to capture accurately. And so this kind of transcription fits in that sense with the human processor model, human computer coupled processor. So what we are attempting to do with the Bell Museum and Zooniverse collaboration falls within the same kind of framework as the Notes from Nature Zooniverse project that Lucy alluded to. That we are taking we're a locally focused approach to this, which will be to some extent distinct from Notes from Nature. So some of the entities that are involved in how this all fits together into a framework, we have the collections, and we have images of the specimens and the labels. We are working with libraries to see that digital information is archived according to a very high standard of digital preservation. The project funded by the state legislature strengthens this piece here, where we are going to present the collections data we have through this Minnesota Biodiversity Atlas, a front end that will serve our databases to regional users like the agencies and the general public, as well as national and global portals where we can publish our data to the world. Where we're excited to be working with Zooniverse is on the transcription of the specimen labels from the digital images that will feed information into the Minnesota Biodiversity Atlas. So if you think about it, we have a lot of great resources at the university that if we can just line them up and coordinate them, we can get some amazing things done, which has its own challenges. But let's just assume that none of these these boundaries around departments and colleges and so forth are rather porous for the time being. So some of the things that we will be undertaking is to pinpoint to the best of our ability the geographic location of each specimen with latitude and longitude coordinates, so that the distributions of these organisms can be distributions of these specimens can be captured through our interface. Through the Atlas, we hope that this is now beyond the transcription. Let's just assume that we are able to do this and effectively engage our stakeholders, like our master naturalists and Minnesota Ornithological Union and other groups, school groups, university courses in capturing these data. What will they be useful for? Through the Atlas, we envision someone being able to draw a circle or capture an area in the state and pull up all the records that are found there, and be able to generate lists of species or drill down to get from scientific and common names of plants and animals down to individual specimen records and their images. To be able to explore explore the database dynamically. Like, let's say these are the distribution of records for a particular species. To be able to click on one, pull up the image of the specimen, get the associated data like Sudan Mine State Park, and then go a bit further and explore dynamically by, let's say, going and seeing, well, what are the records known from Sudan Mine State Park, and pull up a whole checklist for that location. Or using digital images to verify the identity of species in the field, which there are many useful tools for species identification, but generally they do not give you the opportunity to visualize all the variation that exists in nature. So if you can access the museum records, you can pull up, let's say, all the images we have for a particular thistle and look at how variable it is. In terms of charting change where biomes meet, this is the really exciting part, I think, in terms of a research direction that the university community as well as the general public is very keen on, is to look at such things as, for instance, how species geographic ranges are expanding and contracting over short to medium timescales, like let's say the last 100 years, 20 years or so, where these red points represent perhaps recent records, and these black ones historical records. Or to extract other kinds of information from the records, such as biological observations about the ecology of species and their let's say, breeding behavior, or in the case of plants, flowering time, which might be influenced by, let's say, the duration of winter or something like that. And these are some real data drawn from our herbarium specimens for the Juneberry, where over the last 80 years, we have although there's a lot of variation from year to year, we see that in fact the Juneberry is flowering on average 4 weeks earlier in the spring than it did 80 years ago. A very strong response to our changing climate. So this is kind of what we're attempting to do by pulling together support from NSF that supports a lot of the foundational work with imaging of the specimens themselves. The trust fund that is helping us to build the atlas and the kind of the architecture we need to serve up the data, and Zooniverse for helping us find a way to crowdsource the label transcription and really engage the public in doing the science, so that we're no longer behind closed doors, but are actually really in partnership as we move forward in our research. So I hope that this is an example that can demonstrate the kinds of opportunities that you might find with your own data in ways that of engaging the public that perhaps maybe if just a few years ago we never would have dreamed of. So thanks a lot.
I'm Garrett Beier. I'm with the plant pathology department, and I'm working on Dutch elm disease resistance in American elms. And one of the areas I'm looking at is xylem anatomy in resistant cultivars. So one of the many mechanisms that have been proposed for American elm resistance is anatomical. So during the 1970s, they started looking at European elms and Asian elms and comparing those. And Asian elms, if you guys aren't aware, are resistant to Dutch elm disease. And they found that these Asian elms had smaller vessel diameters, which for those of you who haven't had botany for a little while, so these are the xylem vessels here. And then you have fibers here, which I'm not concerned with. But so here you have the xylem vessels, and they had fewer continuous vessels. So continuous vessels would be these ones here that are connected right next to each other and don't have a bunch of fiber cells in between them. So then in the 1975, some work by Sinclair and Associates looked at American elms that were resistant and susceptible and found that they also had this similar trend. However, when they were processing this data, and then in the 70s, they would get an overhead projector and put these thin pieces on there and then trace it on the wall to get the actual sizes. So luckily, we've come a ways from that. But here you can see Princeton. This is one that's fairly resistant to Dutch elm disease, and you also have a wild type. So you can see here it has smaller overall vessels compared with the wild type here, which is susceptible, and also fewer continuous vessels. So here are the continuous. A lot of these larger cells are actually separated where they're not actually touching. So I guess what my work is focused on is I want to be looking at, there's a lot of these cultivars that have been released. So I actually want to compare them to see if they're actually holding up to this and if we can possibly use this for screening them before we actually test in the field for more resistant cultivars. So here's a cross section that I made with a freezing microtome. So for your tree anatomy here, so this would be annual ring 1, annual ring 2, start of annual ring 3. So you have early wood here, which is where these nice large, in the spring you get large vessels forming, and then later in the year you get these smaller vessels forming. So I got these cut and I was all excited, and then I had about 200 of these that I was starting out with. And my first try was I was going to try to use image analysis, and I'll talk about some of the problems I had with that. But then I decided maybe I'll just draw these circles. So I printed them out and started drawing these circles. And about 2 weeks later, I was complaining to my advisory committee, and I said, something's got to change. This is not working for me, and I'm spending too much time on this. So they mentioned the Zooniverse project. So you can imagine going through these, you have hundreds of these images and just lots and lots of circles. So I was circled out. So what I want to gain from this data is different information on the vessels, and then so diameter, shape, area, and then location relative to others, which I can get with image analysis, XY coordinates. And then also for the continuous vessels, I want to know how big each of those continuous vessels are and an average size for those. And then in general, by using the Pazzulli equation, we can figure out, which is the flow through a pipe, we can find out hydraulic conductance estimate on how much is actually moving through the tree. And then also, once we find out the size of these, we can get a distribution of what the sizes are. So, what I first did, how many have used ImageJ? Has anyone done any image analysis? So, great program, love it, but there's some issues. So, for those of you who don't know, when you have an image, you can adjust thresholds, and it can highlight certain areas, and then you can measure those areas. So, I'll talk about some of my problems with that in a little bit here. But another important feature that it has, it's called water shedding. And what it can do is you have these cells. Let's imagine this black line wasn't here. When it draws around there, you can have the computer doing a logarithm, and it'll sense that there's likely a break there, and it'll draw a line. So then this is going to help you separate out these cells. So it can be really helpful when you're trying to analyze this data. So I wanted to just show you kind of the problems that I was going through, just as so you guys can understand what was going through the process. But here's just an image of what I would have initially had. So this is about 250 microns by 250 microns approximately. So this is with thresholding. So what the computer is going to do is I can adjust the threshold, and it's going to measure everything that's red. So you can already see some problems here. One, it's missing spots up in here. It's picking up background noise. I don't want this. And also you have cells that are combined together. So some of that I can fix with water shedding, but it also tends to make lines where they're not supposed to be. And then you have to manually go through and check all of those just to make sure that it didn't make an error. So some of you might say, well, if you've got all these problems with the noise, just lower the threshold. Then the problems get worse. So you can see here, missing out on entire cells. It's not filling the entire cell. So what the main problem is with the thresholding is you get shadows. So you can kind of see the shadows on this one here. And you also get colors that are the same as the background. So you can see the fibers here are almost the exact same color as this vessel element here. So you get all that noise that you're trying to deal with. So what my solution is drawing circles around these, which it comes back around. But I don't have time to do this. So it takes about 20 minutes per one of the big ones to do. So I calculate it takes about 4 months for me to do all of these images. I'll have about 1,500 to 2,000 of those. So it would take quite some time. So once I have this data, then it's as simple as doing this, and it'll give me a count for each one of these, and it'll tell me all of the data that I need from every single one of those. So that's my trials and tribulations with my project. So if you have any questions.
So how many images do you need of one specific plant?
Yeah, so I didn't get into the specifics because I only have 10 minutes, but essentially each tree, I'm sampling 5 different trees based on a power analysis I did. With 5 trees, 2 branches from each tree, and then from each branch I get 3 sections, 3 of those sections, but then I'm looking at rings 2 and rings 3. And when you go back and look at one of the first images I have, some of these are multiple pictures here, so I take them just in order as I see them down. So this might be a 1 picture, 2 pictures, and then a 3rd picture, just to get a single ring. So some of them can have, some of the Princetons that grow really fast, I can have 10 pictures in a single ring. And then that's just 1 ring from 1 tree. So it adds up very, very fast to end up doing those. Yes?
So here, did you have the public then draw circles for you, send them something back?
So what I'm working with Zooniverse potentially on is they would use an elliptical circle kind of shape, and then they would trace the outline of it kind of like I did, only without mine was just with a free drawn, like I just used the pen like I did in Adobe Photoshop, but they would actually be kind of stretching. They'd put a dot down and then stretch the edges to make it fit the size. But it's good to do a trial run on some of these things because you learn a lot more on how long these things are going to take. So, but any other questions? All right. Well, thanks for having me talk too. I appreciate it. So.